So what we'll look at now is the liver. So we'll start again looking at some of those accessory glands that are being encountered on the way. So not actually a part of the tract itself, but it does release some of its vital secretions into the tract, and that has an important role to play in what's happening. So the liver is going to help with absorption of nutrients, right? So nutrients we know don't go, back to the heart. The blood that leaves these GI organs don't go back to the heart. In fact, they go to the liver first to be processed, and then they can make their way back to, then that blood can make its way back to the heart. So the purpose of sending that blood from the GI to the liver first is for processing of nutrients, packaging of nutrients, such as fats and so on, conversion of certain things. So let's say you have excess proteins, you can convert that to some other things. You can deaminate amino acids, so turn that into ammonium and get rid of that from the body. You can turn glucose into glycogen. Just all of that sorting and processing and packaging must take place in the liver. Okay, it also helps to detoxify. So if you're bringing in toxins in your diet, you're gonna have to send that blood, those nutrients packaged together with the toxins to the liver to be detoxified, and then you can absorb more of that, the nutrients there. So this brings us to speaking about the portal system. We would have introduced this system in the lab. So essentially the vasculature that comes from our GI organs that has all the nutrients that have been absorbed must go to the mesenteric vein. So it goes to the inferior mesenteric vein, superior mesenteric vein, the splenic vein joins in, and then all of that converges to form the hepatic portal vein. The hepatic portal vein will go to the liver, taking all that gunk along with useful stuff, nutrients, electrolytes, water, toxins could be in there too, takes all that to the liver, where the liver can sort and package and process that, and then that blood can be delivered to the vena cava via the hepatic vein. So there's a hepatic portal vein, and then there is a regular hepatic vein that goes back to the heart. Okay. We've also got the hepatic artery. So the artery is what's bringing in fresh oxygenated blood that is supplying the cells of the liver with oxygen. Let's talk about the distal parts of the GI tract. So the large intestine and colon. The large intestine consists of the cecum, we've got the colon, and then the rectum. This is going to be the junction between the ileum and the colon itself, where the ascending colon starts. And that region is called the ileocecal junction. This is where we find the ileocecal sphincter or ileocecal valve. And that basically regulates the flow from the terminal part of the small intestine, or the ileum, into the beginning of the large intestine, or the cecum. This is also where we find the vermiform appendix. So the appendix is also found here, and it's also playing an immune role as the contents are coming through from the small intestine to the large intestine. And then we know the parts of the colon, right? There's the ascending bit going up toward the liver. There's the transverse colon. There's the descending colon. Then we get into the sigmoid part of the colon, and then the rectum. The function of the colon is going to be to compact the waste. So really reabsorb, take all that valuable water out of the contents of the colon at that point, and really start solidifying that waste and storing it until it's time to be excreted from the body, a process called defecation. Okay. Just reiterating here, the 4 parts of the colon: ascending going up, transverse going across, descending going down, sigmoid being that S-shaped part of the colon, and then the distalmost part being the rectum. Now the rectum and the anus, this is where we again store feces at the very terminal part of the intestine until it is time to be excreted through the anus itself. This is making up the large intestine itself, so colon, rectum, and cecum, all of that is consisting of large intestine. It's going to have an internal sphincter there that is smooth muscle that we do not control, so we can see that here. That's going to be at the very end of the rectum. But then out towards the end of the anus, we have the external anal sphincter, which is skeletal muscle, and that is more voluntarily controlled in terms of the defecation process. That is what we can relax and allow for that defecation. So the relaxation of both sphincters is required for defecation, but one is involuntary, that's the internal, and then one is voluntary, that would be the external. Okay. All right, let's speak about the accessory glands. So the accessory glands help to produce their secretions via ducts into the GI tract. And again, this is going to aid in some of the functioning of the GI, either movement in terms of lubrication or digestion in terms of the enzymes that are beginning to break these things down. There are 4 glands that we're going to speak about, or 3 rather. There are the salivary glands that secrete saliva. Anyone remember the 3 functions of saliva that we would have spoken about? So we talked about 3 functions of saliva. Anyone can recall those functions? Okay, yep, I'm seeing lubrication, acidity, yep, so it's gonna have the enzymes that help to protect from bacteria. It's going to be involved in lubrication, and it's going to start with the breakdown of food. Excellent. Specifically starch. So breaking down starch or carbohydrates start in the mouth with the influence of the saliva and the enzymes in the saliva. Okay, awesome. And so we're going to talk about the salivary glands that secrete saliva. We'll talk about the pancreas that secretes pancreatic juice. Pancreatic juice is essentially this cocktail of many different enzymes that break down pretty much everything: fats, carbs, proteins, nucleic acids. And then we're going to talk about liver and the acini cells in the liver that secrete bile. Now, acini is basically the part of the gland that actually creates the secretions. So all of these glands have acinar cells, and the acinar cells make up the acini, right? Acinus being a single structure, acini being several different structures. And so an acini in itself is not a structure alone, but it is the component of each of these glands that actually makes the secretion. So for example, if you were to zoom in on one of these acini, one single of these acini, this is what it would look like. This is where those secretions would be made, right? From the acinar cells that are lining this acini, this acinus, excuse me. And the secretions will be made here, and then they're going to flow through toward the duct, and then from the duct, they can be released onto the lumen of the GI tract. Alrighty. Now, as they flow down, they can be modified to be thicker, to be more viscous, or to be more watery, so we can remove water via the duct cells that are lining the acini. We can add more solutes to make it more salty, right? Like saliva is. That is how we can regulate our electrolyte balance as well. So we can definitely modify the secretions that are coming from these cells as it is traveling along the ducts. Okay, some of the characteristics of saliva, just to recap here, we said it is rich in bicarbonate ions. It contains mucus that helps to lubricate the tract. It's got enzymes, so salivary amylase that helps with the digestion of starch or glucose or carbohydrates and start breaking that down into some smaller components. It's also got lysozyme, and lysozyme is an antibacterial enzyme. It is going to help break down bacteria, viruses, other pathogens, fungi that may be present in the oral cavity. And so it's really going to be helpful for cleaning the oral cavity as food comes through and protecting against bacteria or other pathogens. Okay, Second accessory gland here, the pancreas. This is probably the most important accessory gland. It has two different parts. It's got the exocrine part and the endocrine part. We would have touched on the endocrine part in the endocrine system. These are the islets of Langerhans, so these scattered or interspersed bodies of endocrine cells that release their secretion into the blood. So that's the difference between an exocrine and an endocrine gland. Endocrine glands release their hormones into the blood. Right? Remember, we said if it's in the blood, it's going to become a hormone, whereas exocrine glands release their secretions into ducts, right? So no involvement with the blood here. They go immediately into a duct system, and then they are released into the GI. Okay, so we're really focusing here on just the exocrine part of the pancreas. This is going to produce or secrete a cocktail that we call pancreatic juice. It's also rich in bicarbonate, right? And that's going to be important in terms of neutralizing the contents that are coming down from the acidity of the stomach. Relative to its weight, it's going to secrete more protein than any other tissue in our body. So it's very heavy in its involvement with protein secretion. Now remember, all these enzymes that it is secreting are proteins, right? And so it's going to be involved in taking up those amino acids, repackaging them into these enzymes, and then secreting them onto the GI tract. So it's going to be involved more in that role than any other tissue in our body. Enzymes present in this cocktail are pancreatic amylase that help to break down carbohydrates, pancreatic lipase that help to break down fats, proteases that help to break down proteins to amino acids, and then nucleases, which help to break down nucleic acids. So pretty much everything that can be coming through in our diet in terms of macromolecules can be further broken down into their smaller components by the help of this pancreatic juice, which has all these different enzymes. We'll talk more about this system in terms of the bilious tree here, the bilious tree, and how bile is secreted. But in terms of this part, we're looking more at the pancreas, right? You can see the pancreas here, large gland of these acinar cells, right? So we would have talked about the acinar cells that are making up the glandular tissue of the pancreas. They're going to release their secretions. They're going to make their way to the larger ducts. And then from there, they're going to be released into the duodenum, and then they can start breaking down the contents that are coming down from the stomach. And so this is the first part of the small intestine, the duodenum, where most of the absorption and digestion is taking place. Okay, and the third accessory gland here is the liver. The liver secretes bile. Although the liver is bigger, we still want to remember that the pancreas does more of the heavy lifting in terms of its secretions, right? So the liver is bigger, but it only secretes one product, which is bile, that contributes to breakdown. So it's going to secrete that bile. Bile is going to be important in the emulsification of fats. We'll talk about that in the second packet coming up. It's also going to help with, again, processing those nutrients. So converting glucose to glycogen, storing that up, amino acids to fatty acids, which is pretty rare to see that done. But this happens if there's excess amino acids. So the body cannot store away amino acids as easily, so they can convert it to fatty acids or deaminate it and convert it into ammonium and lose that in the waste. It also helps with triglyceride and cholesterol synthesis, and then lipoprotein synthesis. And we need lipoproteins to shuttle our fats around, because we're moving fats around in an aqueous environment. We need kind of this carrier protein to move our fats around, and those are called lipoproteins. Other functions of the liver are part of the catabolism of hemoglobin. So we talked about this life cycle of erythrocytes. And when we break down those old erythrocytes, we're gonna recycle that globin part, and then we're gonna take that heme part and convert it to bilirubin. So the conversion of the old pigment in erythrocytes to another pigment, bilirubin, happens by the liver. Elimination of waste and toxins, right? So eliminating that bilirubin via direct mechanisms, so we can directly secrete bilirubin into the feces, into the urine, or we can convert it into bile. And bile is more useful, again, in the breakdown of fats, which we'll talk about later on, but we need to convert that bilirubin. So bilirubin is actually a waste product. It's actually toxic. And so we have to conjugate it. So a fancy process called conjugation, where we can convert it into bile, and bile is going to be more useful for breakdown of fats. Other things the liver helps with: synthesis of plasma proteins, so things like albumin floating around in our blood, all of our clotting factors, angiotensinogen, which we just talked about in the kidneys. All of those things are synthesized and released from the liver. And then lastly here, secretion and modification of hormones. Again, things like growth hormone are modified and secreted from the liver, and other hormones that help with our growth process as well, so somatomedins, etc. So looking at the biliary system and the biliary tree here, we've got the liver itself. We've also got the gallbladder, and then we've got these associated ducts. So essentially, the liver is what makes the bile. That bile can be stored in the gallbladder. So via the cystic duct, we can use retrograde transport and send that bile down the hepatic ducts, then to the common hepatic duct, and then back towards the gallbladder to be stored. When it is time to have that bile secreted, and the signal for that is going to be a fat-rich meal. The presence of fat in the stomach, as we'll see via some hormones later on, will signal the gallbladder and liver via hormones to release their bilious secretions into the common bile duct, and then that can be released into the duodenum to help with the breakdown of that fat. We also want to think about the duct system. So from the common bile duct, we have the conjoining of the pancreatic duct. So we would have showed the pancreatic duct before, kind of sharing the opening here. And so both of these are joining to form the ampulla of Vater. The ampulla of Vater is going to be the combined joining of the pancreatic duct coming from the pancreas, along with the common bile duct coming down from the gallbladder and liver. There is a sphincter here that regulates the flow from the pancreas and coming up from the common hepatic duct into the duodenum. That is called the sphincter of Oddi. Okay, so the structure itself is called the ampulla of Vater, and then the strip of smooth muscle that regulates the flow of these pancreatic and bilious secretions is called the sphincter of Oddi. Lastly, here we'll speak about the liver itself, so the acinar structures of the liver. So essentially, how do we make bile? We're going to uptake the things that we need to make bile, which is essentially that bilirubin that's coming in from the spleen. So when we process old erythrocytes in the spleen, we send or shuttle that bilirubin component off to the liver. And this is another reason why the splenic vein must go to the liver first, right? So we can take those old dying erythrocytes, break them down in the spleen, and then that blood can go directly over to the liver via the portal system as well. And so the globin part is recycled, the protein part of that. The heme part, we're going to take that, convert it to bilirubin, and then we're going to uptake that in the arteries to the cells of the liver, and the hepatocytes, which are the cells of the liver, can take that and make it into bile. From there, we're going to release that bile onto the sinusoids, and then they're going to drain into canaliculi. So here are the sinusoids, which are forming the lobules, right? So the sinusoids are these channels that are forming the lobules together with the hepatocytes. And then these canaliculi are draining that bile. So as that bile is being produced from the hepatocytes via the sinusoids, we then drain that down into these canaliculi, and the canaliculi eventually converge to form our bile ducts. And then they can then drain that down toward the gallbladder, and then eventually the duodenum. Okay. All right, this is a great summary table just showing you each structure, each organ, and then its function. A good table to start out studying and kind of adding in some more detail. So on top of adding in the function here, or more details of the function, you also want to add some of the mechanisms. So we're going to speak about some of the mechanisms coming up, the enzymes that are involved, the modes of transport that are involved, and you can kind of add those layers into this table.